Thank you very much for inviting me here. This is the second time that we spend time here in this beautiful city. It was an amazing conference the first time, and it seems to be shaping this way this time and even getting better. So I will continue and share with you our thoughts about the impact of trauma and adversity in general on the epigenome. And we mentioned this morning many times the idea that stress is something that we need to consider, for example, with ART. And some games that we are starting to play now, which trying to develop targeted Epigene therapy. So, as we heard today, we should look at epigenetics as the software that programs the cells in our body, and we have to look at it as a network. And what is probably critical is not a single node in the network, but the total output of the network that changes during differentiation. And the idea that we were working on was perhaps the same processes that work during development to confer specific identities to different cell types in the body is also operating in conferring experiential identity on DNA. And the way we look at it is that we are shaped by genetic ancestry through the sequence, and we are shaped by epigenetics through the epigenetics that translates different experiences. And one of the things we were interested in is how those experiences will shape later the phenotype. And these experiences don't have to be limited, of course, to drugs or in vitro fertilization. Stressors that operate in the social economic sphere are probably as strong as chemicals. And this is an obvious idea today, but it took us a long time to get the attention of our colleagues that things like the neighborhood that you were born in could be as influential as the drugs that you have taken or as the chemicals that you were exposed to. And this perhaps, in general, just before I go into my scientific presentation, is a general idea that is not even caught by the general public. And a great example was how we dealt with Corona, where we were focused only on counting infections, but not focusing on the socioeconomic implications of our decisions, and which might be much more harmful than the initial insult. So when we go to trauma, this is our working hypothesis. Early exposure to stress triggers alterations in DNA methylation in the brain and systemically. And I want to focus on the fact that the brain is not floating in air, as some psychiatrists think, but the brain is connected to the rest of the body. So therefore, even stress that is behavioral is registered in other parts of the body. The readjustment is activated by stress response hormones and other hormones, such as glucocorticoid receptor. And this methylation matrix that is formed early in life defines the response to trauma later in life, whether through resilience or PTSD. One of the interesting things about PTSD is that you can have 50 soldiers in the same battle exposed to the same traumatic experience, but only a certain fraction, 17% or 8%, depends on different societies, will develop PTSD. So the same environmental signal, but a completely total different outcome. And trauma-altered methylation then will define resilience or susceptibility. And of course, as a pharmacologist, PTSD could be reversed by appropriate epigenetic manipulation. So this is the working hypothesis. And I'll describe to you one model that we studied in the last 8 years. And it is a rat PTSD model that was developed by my friend Gal Yadid at Bar-Ilan University. And I especially like this model because, in difference to other models of a conditional fear, it recapitulates what happens in humans, which is the greatest trauma for a rat is a cat. And let's think about it. All the rats that are not afraid of cats became dinner during evolution. So all the rats that we have today know how to be fearful of cats. So the fear that developed to cat is an adaptive, of course, trait. And if you expose a rat to a cat in a certain context, and then expose the rat to the same context without the cat, the rat will develop symptoms of PTSD, like PTSD, which is measured by different behavioral phenotypes like hyperarousal, freezing social interaction, and freezing exploration. So you can measure that. And what you see is that following the reminders, you can measure those freezing events, and you can see clearly that there are not two groups, but three groups. One group, of course, is the control that was never exposed to trauma. One group was exposed to trauma, but is resilient. And the other group was exposed to trauma and develops this freezing reaction, these three behaviors. So that was really interesting to address from the point of view that experience will not have the same impact on everyone. And it was a huge opportunity to study. But by the way, this took a long time to realize that that's the case. And many behavioral biologists notice this phenomenon that they do an experiment and there's a huge standard variation. And usually we kill the experiment because the standard variation is so big, so no statistics, especially with a small number that you do in an animal experiment, will see anything. But once you break it, you see clearly there are two groups. There are those who are exposed to trauma and they're absolutely normal, and those exposed to trauma and lose it. So that's an opportunity to ask the question, what's the difference between those exposed to trauma and those that lose it and those that are susceptible and are resilient? And perhaps if we find that, we can start thinking about how to fix it. So when we mapped the methylation in the nucleus accumbens, which is a region that is responsible for reward in mice and humans, we saw that there are, of course, DNA methylation differences between susceptible and control, between resilient and control. My mouse doesn't work here, right? So we don't see. And there were unique differences between susceptible and resilient. So you can see that there's an epigenetic response to trauma, which is common to resilient and susceptible. So it's not that the resilients are completely controlled, they didn't see anything. They develop an epigenetic response, but there's a unique response for susceptible. And you can easily cluster the animals between those who are susceptible and those who were resistant. So the first assumption that there is going to be an epigenetic difference between resilient and susceptible seems to be supported. So we try to understand what is the source of this difference, and we found that the overall DNA methylation level, not just site-specific, are different between the controls and the susceptible and the resilient. And when we try to look at the enzyme that is very much responsible for de novo methylation in the brain, DNMT3A, you can see that susceptible animals have a reduced activity of DNMT3A. So the methylase seemed to be kind of induced in the resilient and perhaps offer them protection. And you can see, of course, that there is a nice correlation between how much mRNA they're expressing and the methylation levels. But also, interestingly, there's a correlation between the amount of mRNA of DNMT3A in the brain and the freezing behaviors, and the three freezing behaviors. So the response of DNMT3 RNA to the trauma is variable, and the susceptible are expressing much less methylase than the resilient. We also looked at the pathways, because if I started with a picture of network, right? So we wanted to see if there are some upstream regulators that are different between susceptible and resilient. And two guys came to shine, and they're all involved with retinoic acid. Interestingly enough, one of the genes that we found, RORA, was also found in GWAS to be associated with PTSD. And by the way, one good avenue that we have is to see if our methylation hits are picking genes that were also detected by genetics. So now we have two candidates, retinoic acid pathway and DNMT3A pathway, and we can ask the question, can we rescue, can we cure those rats by injecting DNM3 RNA and retinoic acid RNA, RORA, the orphan receptor for retinoic acid, into the animal's nucleus accumbens. This will do two things. First, we'll establish that these probably play a causal role in the phenotype, and the nucleus accumbens is important in the phenotype. So when you look at the animals and you compare the resilient to susceptible, and remember, the higher it is, the more PTSD they have, because we are measuring how long these animals are freezing in these three different behaviors. So these are the resilient, these are the controls. So the controls show this freezing phenotype in the three behaviors. And when you inject the DNMT3A, you can see that the controls are becoming almost like the susceptible become like the resilient. You reduce the difference between susceptible and resilient by injecting either the DNMT3A or retinoic acid orphan receptor RNA. And you can do the opposite, which is, can I now turn the resilient animal to become susceptible by knocking down DNMT3A and RORA? And you can see that indeed this has happened, that the freezing goes up when you inject to the resilience DNMT3RA or retinoic acid RORA shRNA. So if you upregulate DNMT3A or RORA, you cure the phenotype, you make the susceptible resilient. If you knock down DNMT3A or RORA, you turn the resilient into susceptible. So that also establishes that these phenotypes are reversible and potentially amenable to treatment. So if we look at epigenetic states as some sort of a network that now has a different output, can we reverse the network to the other side? And we can think about two strategies. One strategy is a selective strategy. Let's identify the important agents in the network and direct targeted therapy to these targets. The other approach is the approach that I have with my computer. For example, I have a Surface computer, which is cool because it has a Surface Pen, but it stops working every 15 minutes. So what do? I power it off, I power it up, and it works again. So I essentially completely disrupted all the stuff that is going on without knowing exactly what I did, turned it on, and now it works. So this is one idea of epigenetic therapy, and we try to mimic what we saw by giving them nutritional supplements. The idea of nutritional supplements is that here we have non-toxic agents that could be easily translated to therapeutics without having to go through a billion-dollar drug development process. So we injected two agents, acetylmethionine and retinoic acid. And what we saw is that the effects were almost very similar to what we saw with injecting the genes themselves. So we could reverse hyperarousal with retinoic acid and a combination of retinoic acid and SAM, but interestingly not with SAM. But we reversed the social interaction freezing with both agents and exploration with both agents. So when we injected retinoic acid and SAM, the animals that were susceptible are now not different anymore than the controls. And the other thing about the treatment that we did is we tried to mimic a treatment that could be done in the psychologist or psychiatry office, which is the way, the psychiatrist is now treating PTSD. One of the most successful ways is through re-exposure therapy, which is they start showing them movies about the trauma and talking about the trauma. It's essentially a re-exposure that works, the only treatment that actually works with PTSD. And we combine, so in the rat, we mimic the psychologist's office by giving in the bedding that saw the urine of the cat. This is the re-exposure therapy. And at the same time, we provide the drug. So we provide the drug once or twice, and we get this kind of response. So to summarize, the working hypothesis is that lifelong trajectories that are defined by early adversity set the stage for different responses to trauma later in life. They create the inter-individual difference that now will create a completely different DNA matrix. And when the trauma hits, some people will become PTSD and others will become resilient. And the question is, could we use the strategy that we used in the rat to mimic it in a clinical setting of treating PTSD? So this is a very crude kind of treatment. It's not very elegant. You're using a general agent like SAMI, which you can buy for $34 on the internet. And you don't need a doctor. You don't need anyone to use it. But of course, it's not very attractive, right? So the next question we asked is, this is a sex gender specific meeting, so I had to talk something about sex. And first, to confess that we committed the crime of only treating male rats. And the reason we committed this crime is because we didn't have enough money to do both male rats and female rats. But we went deeper into the idea that a glucocorticoid receptor plays a critical role in shaping the methylation pattern. And we looked at knockouts of heterozygous knockouts of glucocorticoid receptor and looked at DNA methylation in the placenta of these mice. And what we saw is that if you compare, and this time we compared males and females. So if you just look at the placenta of wild-type males and females, we see very little differences. If you look at wild-type versus the heterozygotes in females, you see very large differences in methylation. So the hypothesis that the amount of glucocorticoid receptor plays a critical role in shaping the DNA methylation pattern is supported. But look at this. When you look at the males, you see almost an opposite pattern. Like you see hypermethylation versus demethylation. And when you compare now two animals that have a genetic difference in glucocorticoids, now the difference between males and females is very, very large. And this is a very nice example of gene by sex interaction, where the same genetic deletion has a complete, almost opposite effect as far as methylation pattern in males and females. So these mice are now, we also stressed these mice and did all the things that one should do with them, and hopefully one day we'll analyze the data. We have the data, but we haven't fully analyzed it to see the sex effects on this response. So we have kind of genetic evidence that glucocorticoid receptor plays a very critical role in shaping the DNA methylation pattern, and that this is done in a sex-specific manner. But one of the challenges of DNA methylation is moving from causation, from association to causation. Proving causality is very difficult. So in the experiment I showed you, we did prove causality, but not for DNA methylation. We use DNMT3A. Yes, it is an enzyme that methylates DNA, but it does also other things. Yes, we treated with SAMI and reversed the phenotype. SAMI indeed contributes to DNA methylation, but it also contributes to histone methylation, neurotransmitter methylation. It's not a clean thing. And even if we just change DNA methylation, we don't know whether the sites that we identified have any role, or they're just the fingerprints of something else that is happening and plays no causal role. And this has plagued the field of DNA methylation for the last 40 years that I have been investigating it. So we decided to do something about it, and we found out that CRISPR editing through steric hindrance could provide us with very site-specific DNA demethylation. And we took one gene in the mouse fibroblast. This is the interleukin 33, which we found to be methylated, and we found that it could be demethylated with AZA-C. This is kind of a proof, and induced a little bit with AZA-C. And this is what we used as a proof for DNA methylation regulation, but it really isn't, right? Because how do you know if AZA-C activates the genes through IL-33 methylation? It also activates SP1, it also activates all these transcription factors. So when you treat with AZA-C, you really don't know whether the methylation event that you are measuring the changes is causal. It might be a total bystander because AZA-C might be doing something else. So then we went to the accepted CRISPR methodologies to change methylation. So one method that is very common now is tethering TET, which is the enzyme that oxidizes methylcytosine, eventually leading to its loss of DNA methylation with delta-Cas9. So you can target it, and then the TET can demethylate and then oxidize, and then there's another set of enzymes that come in and demethylate. So this looks good, but we found many problems with this method. First, essentially we're expressing huge amounts of a TET enzyme. It's true that it's guided to a certain site, but nothing stops this TET from going to other places in the genome. The fact that you target it doesn't mean that it doesn't go untargeted. And we found out that TET can cause activation even if it's mutated. It doesn't cause oxidation. So TET has other activities. By the way, this is a warning site. No protein has the activity that it was discovered to be with, right? So every protein can do multiple things. So if you just knock down a protein, you proved that the protein is important, but you cannot prove that the biochemical activity of the protein is important because there are other activities that the protein does. So we found out that this doesn't work. It doesn't give us a picture of direct demethylation. We also found out, as I said, the TET activation of transcription with TET dead mutant and non-specific activation of distant promoter, and I will show you later that TET activates thousands of other genes because you are essentially loading the cell with an enzyme, hoping that it will go to your target, and it does, but it also goes everywhere else. So we developed this following method. And this came out of our frustration because what we saw is that our TET-directed CRISPR editing was not what we wanted, but we noticed one thing. TET completely demethylates at the gRNA sites. And this has nothing to do with the TET activity. And we understood that what's going on here is, what are you doing? You're taking a gRNA, guiding it to a specific place in the genome, and Cas9 is sitting on it. It's a delta-Cas9, so it's a dead protein, but it's just sterically inhibiting DNA methylation. And that could be a method that is clean from other enzymatic activities, so we can start asking how specific sites are participating in DNA methylation. And you can see how specific each of the guide RNAs are. So for example, this guide RNA demethylates these 3 CPGs, this guide RNA demethylates only 1, and this demethylates the transcription start site, which we then went to examine our IL-33, and we could show that using luciferase activities, that the unmethylated plasmid is active. This is the gray. If you look at the methylated in black, it's silenced. And if you look at the red column in the bottom, this is the gRNA that demethylates the targeting sites, actually prevents methylation. And so there's a problem here, because if the DCAS9 sits on the gene, not only it prevents methylation, it also prevents the binding of transcription factors. So again, you're convoluted. You don't know if it's transcription factors that are prevented in binding or demethylation. So you follow this by, we used a site-specific recombination with CRE, with a CRE-LAC system. So you can put the delta Cas9, you put two CRE-LAC sites next to it, you demethylate, then you remove it, which is very clean because now you don't even have the Cas9, you have an unmethylated site, and now you can, as you can see here, it's totally unmethylated, and you can ask the question what role it plays in gene activation. And what we found, to our surprise, not the big role. The demethylation of transcription star site had a small effect on expression of the gene, but not a huge effect. And this is just to tell us why the DCAS9 with TET cannot work. Yes, it is targeted here, but it also acts on many, many other sites. So we did genome-wide sequencing of the DNA methylation, and you can see that with TET, you demethylate huge amounts of sequence. So I suggest that you can revisit all the literature that has used this method to show site-specific demethylation and activation of genes. However, we find one gene that is dramatically activated by demethylation. And this gene is the FMRR1, the fragile X gene. And when you take fibroblasts from patients that have the FMRR mutations, this is, you know, a CCG repeat disease. It causes very strong activation of the gene by steric hindrance inhibition of DNA methylation. So we also need to understand that activation of a gene, so why FMR1 worked, and other genes we got very small effects, and the reasonable explanation is activation of a gene might require a combined demethylation of several genes. So indirect activation of a methylated gene by demethylation of different genes, right? So for example, we have this gene, oh, the mouse doesn't work, okay. That is demethylated, but it's not activated. And the reason is it needs a transcription factor. So you need to demethylate another gene that is the transcription factor of that gene to get full activation of that gene, which implies that if we want to do targeted demethylation therapeutics, we will need to do more than one target because we will need to also demethylate possible regulators of the gene. Another interesting point is the idea of anticipatory activation. A common mistake that most people do is to correlate DNA methylation with expression. But what they are doing really is correlating steady-state mRNA expression with DNA methylation. And if indeed all the role that demethylation has is to be like expression, there's no reason to measure both. You can measure one and that's sufficient. But the same idea is that a gene needs really multiple events to be activated. So when you demethylate a gene, you essentially make that gene ready for the time that the towers will be attacked at 9/11. So the guys that had the genes demethylated activated it with the trauma, versus others that didn't have it methylated didn't. So up to 9/11, there was no difference between these two people. But the anticipation that was programmed in the demethylation changed the phenotype. That's why many times when you measure methylation correlation with phenotype, you have to measure it under some sort of an activation state. And the best example is the glucocorticoid receptor. So a student of mine, Nadine Provençal, showed in her own work with Elizabeth Binder a beautiful idea where she was looking at activation in precursor neuronal cells by glucocorticoids. And what she showed is that demethylation of glucocorticoid responsive genes only has effect when you later treat those cells with glucocorticoids. So the cells that had the FKBP5 demethylated will react differently than cells that didn't. This is exactly what we're talking about. Like these two people were similar till they encountered trauma, which is mimicked here with a firing of glucocorticoid. So that means targeted demethylation treatment could not have an effect, immediate effect, but would have an effect if that person is facing trauma later in life. So early on, there's no difference. This one is demethylated, this one is not, no difference. But when the HPA axis is activated and there's trauma and there's a firing of glucocorticoid, this guy will activate the gene, this guy will not. So what are the prospects for epigene therapy? You know, we're talking a lot about gene therapy. What is the prospects for epigene therapy? And could we actually address epigene therapy in the same way that we address gene therapy? So we need, first of all, to know which methylation sites have a functional impact on disease. Oral literature, almost all of it, is correlational. And we get very little idea about which DNA methylation sites are important. But we have few candidates. For example, tumor suppressor genes, differentiation markers, immunoregulators. We can activate alternative silence pathways in genetic deficiencies, like, for example, in thalassemia, mental health disorders, induction of insulin in diabetes, induction of stem cells. So there's a whole panel of targets that should be amenable for epigene therapy. However, we have challenges. The beauty of gRNA approach is that you can actually send multiple gRNAs to a cell, and you can actually do what I described before, which is knock down not just one gene, but multiple genes. So that's doable. And the other thing we showed, that it is a hit and run. So if you just block it, you don't need this thing anymore. So you can use essentially RNA as a drug, right? Because you don't need this RNA to stay for too long. Actually, you don't want it to stay for too long. You want it to go in, to do the job, and eventually be diluted and eliminated. And what we need is to solve the challenge of RNA protein delivery. And this is a challenge, of course, that a lot of companies are working on with gRNA CRISPR approaches for gene editing. But I think for epigene therapy, it will be much more useful, much more drug-looking like, and hopefully with the development of RNA modifications and RNA stabilization lessons that were learned from RNA vaccines, this idea of using RNAs as treatment will become much more feasible. And actually, Moderna has now established a whole RNA research facility in Montreal to look at the potential of RNA as drugs, using the vaccine model as an example. So I will finish with acknowledgements to the colleagues who helped with the work. And again, thank you for offering me this spot to share these thoughts with. Thank you.